Good morning, hires. And today what we're going to do is we're going to look at key area 6 of Unit 1, which is mutations. Now, you might have heard of mutations before, but we're just going to break it down. So this is a simple definition of mutations. Mutations are changes in the genome that can result in no protein or an altered protein being synthesized. So we have just been looking at protein synthesis, looking at processes such as transcription, uh, RNA splicing, and translation. And now we're going to think about mutations. And mutations are random changes to the DNA. So they will occur randomly, but they do occur. And we're going to look at different types of mutations. And the first one we're going to look at is this thing called a single gene mutation. So as it suggests, this is only happening with one gene in the DNA molecule. So it's a change to one gene. It involves the alteration of a DNA nucleotide sequence. So think about back to thinking about the DNA molecule. It's made up of these individual nucleotides. So it's going to be a change to this, one of these nucleotides. And this can be due to uh, one of these three things. So there's substitution mutations, insertion, or deletion mutations. So a substitution, insertion, deletion of nucleotides. We're going to look at each of these individually and think about what they are, and why they can have a significant impact on protein being produced. So the first one we're going to look at is substitution. And the clue is in the name. So if you think about a football team, if a player gets substituted, one player comes off and a new one comes on the pitch. And it's a similar idea here. One base is substituted for a different base. So say you had thymine, it could be substituted for guanine. And we'll look at a couple of examples of this. Because it's only one nucleotide that's getting changed, it only has a minor change. So you're only changing one base. So this usually only has a minor change to the protein being produced. And it usually results in only one different amino acid. So if we have a look here, we have our DNA molecule. We have our DNA bases. And remember, from what we've done before, a triplet of bases, so three bases, is one codon. There's another codon, and remember, each codon will code for one different amino acid. Okay, so they all code for this amino acid. Now, if you were to then substitute, say, this base here, this adenine, and its changes to this one, which is cytosine, you can see that this is no longer CAT, this is now CCT, and so this will code for a different amino acid here. And so you can see this is the incorrect amino acid, which may produce a malfunctioning protein. So because one amino acid is changed because of the substitution mutation, it means that the whole protein might malfunction. It might not work properly. Now, we're going to break it down a little bit further because there are three main types of substitution mutation. We've got missense, nonsense, and splice site mutations. And these occur when one single nucleotide is substituted. So we're breaking it down even further. And we're going to look at each one individually. So if we look at a missense mutation, after mutation, the codon codes for a different amino acid. Okay, so that's similar to the one we've just looked at with this example here. You've changed one base has changed with the mutation, and therefore a different amino acid has been coded for. That would be a missense mutation.